Okay. Well, thank you for the introduction, and I'm really happy to be speaking today for the European Polymer Conferences. I changed the title a little bit because I think it is polymers, conferences. So I really want to highlight how use chemistry are important to design the materials, properties. And then today, I really wanted to showcase that in my group that how to really using simple chemistry that can really design really complicated the microfunctions, multifunctions and microstructures, the biomaterials, and then really sort of hone in this concept, how simple is complex enough in terms managing of, platforms to promote cell functions and that sort of towards the tissue engineering applications. Um, So my lab is really a bunch of bioengineering, chemical engineering people. We're trying to make biomaterials to grow tissues. So we sort of generate this hydrogel-based scaffold materials at the center of our research portfolio. Under the principle of tissue engineering, we really use, a selection of materials and also the different chemical cross-linking method to enable the encapsulation of different cell types in these scaffold. And on top of that, we can apply different technologies and build these cell-laden cell constructs that can use for patterning different tissues as well as achieve the tissue functions for tissue engineering applications. So really, we're looking at combining cells, materials, and engineering approach, which is the core of the tissue engineering principles. So overall, this is the sort of a portfolios, what my lab's really trying to do. I'm sorry, I forgot to introduce. I'm actually assistant professor from University of New Hampshire in the Department of Chemical Engineering and Bioengineering. So my lab research really at the interface between material scientists, material science, and biomedical engineering. We focus today, I'll be talking about really how to focus on developing novel biomaterials with unique biophysical properties and use this as a platform to control and understanding the biochemical signalings and then integrate that into the in vitro system with the synergy of biochemical and biophysical signaling to control the cell functions. And then we use this mostly for our applications, our chronic wound healing as well as angiogenesis, which is a part of the chronic wound healing sort of process. Given this community is more of the sort of on polymer side, I wanted to give some sort of background on tissue engineering, particularly on the wound healing process and sort of how we sort of motivated by the physiological process of these wound healing and then using that information to guide the design of our material. So with the aging populations and increased number of the diabetic patients, so the Medicare data indicate about 7 million patients alone in the US are associated with the chronic wound process. And then the estimated cost of treating these chronic wounds is estimated to be 25 billion annually. So this is really becoming a hidden epidemic and also worldwide situations that needs a lot of innovation in solutions and come up with the treat these issues. So typically for wound healing, we are typically categorized in four distinct stages. So you have the hemostasis, which is the stop bleeding. So we have the fibrin clot formation at the first stage. Then immediately you're followed by the inflammation stage, which is to have the macrophages neutrophil infiltration to clear the wound bed, and then lay down the cytokine to trigger the migration and proliferations of the fibroblasts, then followed by the proliferation phase, which you form the granulation tissue formation, which is the pink tissue area before your wound actually heals. So this is a critical area, a stage, because now not only you have the granulation tissue formation, you also have the characterized by the wound contraction and reepithelialization, and then finally you have the remodeling stage, which is continue to reconstruction the ECM molecules as well as the myofibroblast maturations, and then you have a physiologically healing process. So as I mentioned, the third stage, which is proliferative phase, which is critical phase that bridge to your inflammation to the final healing is essentially for your rapid wound healing. A lot of the chronic wound healing is because you can't reach to this phase or stay in this phase really long that prevent the healthy wound healing become the chronic wound healing. So this phase that features the granulating tissue, which is characterized by a dense vascularized provisional tissue that contains multiple different cell types as well as cell-related ECM properties. So not only have the sort of proper cell types as well as the sort of amount, the matrix that deposited by the cells. So this is critical steps that moving into the sort of a healthy and physiological wound healing processes. So the challenges in healing these sort of chronic wounds, we categorize in these different bullets. I think this is important for us to think about what is the issue in the biological events that guide us to design our material can be really effectively to actually, healing the process. So here we have a couple things I think it's really important to point out. The first one is prolonged inflammation phase, which is that sort of a persistent infections, that biofilm formations, so they can actually prevent moving along to the different phases, which is mess up with the timeline. And then the second part is the decreased concentration of the pro-angiogenic growth factors, for example, VEGF, BFGF. So now with the sort of a lower amount of these critical growth factors, now you fail to induce the angiogenesis. So these we are considered about the biochemical signaling. And then because you have lower concentration of the gradient, the growth factors, now you are not successfully to induce that progenic endothelial cells to move into recruitment. So now this is because of biochemical signaling, you lack of the cell sources. And the other part that we also thinking is critical, which has been less emphasized in the tissue engineering, which now gain a lot of attention, is at the chronic wound bed, you have excessive amount of protease activities, that overexpression of the metalloprotease enzymes that constantly degrade the matrix. So whatever cells produce, synthesize newly deposited ECM just get degraded. So now not only you don't have the buildup, and not only that you lacking of these, you also constantly lower the mechanical properties as well. So we consider that as the biophysical properties. Now, because your ECM molecules that gets degraded, so the cells cannot effectively to migrate. So like, for example, fibroblasts and endothelial cells. So now you not only lack of adhesion, you also inhibit their sort of migration excessively, which required for the wound healing closure, so that delay the epithelial migration, reepithelialization, which is the end of the wound closure. Again, then, all these can be, categorized in classical looking at biochemical and biophysical, but I think that how the biophysical environment changes is really impacted by chemical signaling, which is we trying to sort of using our system to understand the properties. So we are particularly interesting to really understand and engineer the sort of materials that mimic extracellular matrix. Extracellular matrix is the materials that's sitting outside of the cell, outside of the cell. It is a heterogeneous and hierarchically structured materials that contains very different molecules. For example, your collagen, you have your glycoproteins and fibronectins, your basement membrane protein, laminin, and proteoglycans as a GAG system. So these sort of a heterogeneous chemical composition really controls a lot of chemical signalings that support cell functions, survival, adhesion, as well as differentiation. At the same time, they also build up these sort of mechanical support for the cells. So because the organization and composition that with the cell type is specific to the tissue, that gives the variety of different sort of mechanical differences to your specific tissues. However, currently, the cell culture platform that we still using are extremely stiff sort of tissue culture plastic platforms. Not only you cannot tune these sort of mechanical properties to match with the specific application you're trying to do, but also just doesn't really mimic the tissue environment. As we know, the pioneer studies from Dennis Disher lab that shows that the cells can differentiate and then depending on their environment, basically on the stiffnesses. So the first question we're trying to ask us is, can we establish a synthetic polymer base? We're focusing specifically on polysaccharides. that not only allowed you to actually to mimic the sort of a native tissue mechanical properties, but it also allows you to actually using these ECM proteins, not through the chemical conjugation, as we know, when you chemically modify using these ECM proteins, you might change their bioactivities. So we wanted to develop a simplest platform that allow you to tune the mechanical properties and also absorb the ECM proteins that so can actually using that to investigate how the biochemical signals as well as the biophysical properties that impact the cell adhesion and functions. So to do that, we sort of looked at a lot of candidates in the material category, and then we decided using dextran, which has been widely applied in the biochemical, biomedical field and material science community as well. So dextran is a linear homopolysaccharide. It only has one repeating units. Repeating units is glucose, so it is hydrophilic, it's non-charged, it's the glucose monomers, and it's very cheap. So I list a couple other materials that currently widely used as the biomaterials: HA, PEG, fibrinogen, and collagen 1. So all these materials, you can see the prices as well. So you can do, because it's a homopolysaccharide, so it allow you to do the large quantity processing and then has really reliable sort of a repeatable results for the functionalities you achieve. So it is also biocompatible, it's bioinert, has no known cell surface receptor, it is also FDA-approved polymers. We consider the dextran is the sugar version of PEG, as they only have the hydroxyl group as a functional group. So the first question we trying to sort of address is how to develop a hydrogel platform to using this material. So to that, we sort of resort to the literature, which has been widely used as these modification of the backbone through the hydroxyl group and then modify and with the methacrylate groups so through the one step modification. So now, once you go through this modification, you can achieve these methacrylate modification of the dextran. So now, and then because they have these non-saturated double bonds, we can use the photocross-linking to form these hydrogel. So, and then turns out when you actually changing the modification of the methacrylate from the by changing the GMA and as well as molar concentration of the GMA to the dextran units, you can change the methacrylate functionalities. From lowest one is about 17% all the way to about 88%. So it turns out that the degree of the methacrylation functionality really controls the hydrophilicity of the hydrogels. So the hypothesis, when you modifying the glycerylmethacrylate modification gets the methacrylate modified, you changing from relatively hydrophilic, which give you the hydroxyl, which forms the hydrogen bonding with water environment. Now you modify them with the methacrylate. At this conversion, if you gradually adding more of the methacrylate to the backbone of the dextran, you actually become from a hydrophilic monomer become a hydrophobic compartments. So at a certain point, we'll see that when it gets to the higher modification, you can generate a relatively hydrophobic molecule of your modified dextrans. And then it turns out that once you mixing the different ratio of the methacrylate, you can either control them swell. So if you look in the dotted line, which is the dimension of the hydrogel before you swell. And then if you mixing the different ratio together, you can actually control hydrogel non-swelling. Because I think right now, the major issue that for the hydrogel community is that when you're making hydrogel that swells significantly, over the course of the cell culture, you're actually changing not only the pore size, the diffusion of the nutrients, as well as the mechanical properties. So you don't have the original properties you want to achieve, and then the final properties is significantly different. So what we wanted to do is actually generate hydrogels that has minimum swelling or non-swelling hydrogels that maintain not only the mechanical properties, but also the physical structures. So now we see from the figure here that we're able to achieve the high modification. And then if you see this sort of a different, the yellow, orange color circle as well as the light green. This is a 70% modification, which is we saw is a threshold that above the 70% modification that you actually generating a phase separation. So the below 70%, you actually can have the dextrin solution dissolve that without phase separation. But at once you go higher than 70% modification, you can trigger a liquid-liquid phase separation at room temperature. So that for the first part of the talk, I'm going to focus on everything that below the 70%. So now we actually, in order to quantitatively to characterize the swelling, the hydrophobicity of the hydrogel, we're using UV to crossing hydrogels, and we're using contact angle to quantitatively analyze how the contact angle associated with the methacrylation. As you see that once you increase the degree of the methacrylation, you also has the a gradually increase in the contact angle, which above 80 degrees of the contact angle, we consider them as a hydrophobic hydrogels. So now the next things we want to do is to see if these highly hydrophobic hydrogel surfaces can absorb the ECM proteins that we just basically dissolved in the PBS to the surfaces to show that we can have this non-specific sort of absorption and then promote the cell adhesion. So the sort of flow is that now we have the protein in the solutions. We incubated at 3741 degrees, and we hypothesized that with the hydrophobic surfaces, we're able to absorb, pull down the ECM protein onto the surfaces, and then actually these ECM proteins can promote cell adhesion. So the first thing we did is we're looking at using a fibronectin as a model protein, and then we just do the process and looking how the ECM proteins can actually be absorbed on the surfaces. So now you can see that F is indicating the modification, theta is indicating the contact angle, and then you can see there's a direct correlation between the modification of the methacrylation as well as the contact angle. As you increase the contact angle, you have the increased amount of the absorption of fibronectin. Of course, we see some of the sort of a hydrophobic mediated aggregation of the fibronectin, and then we quantified and then see that there's directly increase as you increase methacrylation, you have more of your fibronectin absorbed on the surfaces, right? So this proved that we're able to engineer surfaces, that hydrogel surfaces, hydrophobic enough that actually pull down the ECM proteins in the PBS sort of media. So now the question is, can these proteins able to support cell adhesion? And then we then seed our cells onto the surfaces. As you can see, there's also direct correlation. As you can see, the cells, we're using fibroblasts as a demo cell types. You can see there also a direct correlation as you have significant amount, which is correlating here at the highest the functionality of the methacrylation and also the contact angle, you have more cells, not only more cells able to adhere to the surfaces, you apparently have the larger cell spreading as well. So we quantify the cell area as you show there's direct correlation as you increase the methacrylations. You also have a larger cell area as well as the back ratio, which is the length divided by the width of the cells, as the higher aspect ratios indicating the cells more elongated. And then at the highest modification, you can see this fibroblast showing these sort of a spindle morphologies. So we confirm that now we have the hydrophobic substrates that can not only promote the ECM protein absorption, but also the absorbed proteins can actually support cell adhesion. So again, then the next question we want to ask, is this specific for fibronectin, or can we actually using these more generalized to a variety of different ECM proteins? So we're now using the commonly applied 6 different type of ECM proteins: laminin, collagen 1, collagen 4, matrigel, fibronectin, and fibrinogen. You can see, and this is SEM images to see that it is showing you that we have the different morphologies and actually capture some of the native ECM sort of a morphology that have been able to absorb onto the surfaces. So this is really interesting to us that we can actually generating more generalized substrates allow to actually absorb a variety of different ECM proteins. Now, the next question we want to ask is, are these different ECM proteins can be sensed by the cells and show the different morphology? Then we actually again see the human dermal fibroblasts on the different surfaces and show that that we do have the ECM specific cell type morphology. For example, the fibronectin again shows the spindle morphology, and the laminin you can see that's really long and thin, have these major protrusions that's shown in the typically on the laminin surfaces, as well as really recaptured by the matrigels, as matrigel, the main component of the matrigel is the laminin. So, and then collagen 1 is not because the triple helical structures is not the well defined cell morphology in these cases. And then we quantify again the cell area. You can see the laminin, the collagen 4 has a larger cell area, as well as in aspect ratio. You have the laminin and the matrigels had the highest sort of aspect ratios compared to the other different ECM coated surfaces. So now this is again to show that we're able to develop these hydrophobic hydrogel platform that can be applied for a variety of different ECM protein absorptions and show that we have achieved the ECM specific protein morphologies as well. So now this is sort of considering if you can able to show that we can change the different ECM proteins that we consider as a biochemical signals. Now the next question, can we decouple the biochemical and versus your biophysical properties? As we using the UV to crossing the hydrogel because it's radical polymerization. So now we're able to tune simply by the UV radiation time and then control the stiffness, which shows 30 seconds to 120 seconds. We can achieve the lowest stiffness is 1,000 pascals and the highest about 10,000 pascals. So we can tune the mechanical properties. And then interestingly, when we actually change the mechanical properties from soft to stiff, and then the contact angle doesn't change. So all the contact angle, so these hydrogel made at 70% modification. So all the contact angle actually doesn't change in here, which indicates that you should not have they have the similar hydrophobic behavior. And then because the contact angle didn't change, you should not expecting anything changes in the swelling ratio. So we actually showing that these is essentially non-swelling hydrogels. You can see that we can decouple the stiffness from the non-swelling behavior. So now not only are we able to decouple that the mechanical properties from the biochemical signals, but within the mechanical features where we are actually can faithfully say that we're able to change the hydrogel stiffness that generating non-swelling hydrogels that have the different stiffness. And then using this different stiffness, again, we're using fibronectin to absorb on the surfaces. You can see that now using different stiffness, we have the similar amount of the fibronectin absorption, but also the quantification here as well. So which to us now can really decouple the biochemical now. So biochemical signaling is consistent because we have the similar amount of the fibronectin absorbed on the surfaces. And then the determinant of how much fibronectin absorbed on the surfaces is not by the mechanical properties, is by the hydrophobics of the substrate, which is determined by the contact angle, which is further determined by the methacrylation degrees. So now we generating a sort of a category of the hydrogel surfaces can really decouple your biochemical signals away from the biophysical parameters. So now we're using these sort of behavior to really to understand how these are changing the stiffness and regulate cell behavior as well as at the simultaneously changing the biochemical signals. So here we show a classical sort of adhesion impact on a mechanical impact on cell behavior. We're using the human dermal fibroblasts as one example, and then using the human umbilical vein endothelial cells, another category cell type. So here we can see that in the fibroblasts, we have this classical, when you increase the stiffness, you show the increased sort of cell morphology, as well done and shown that by many other labs and pioneered by the Dennis Dish's paper, how stiffness regulates cell morphologies. Again, and then HUX, which is interesting, we see the behavior, but I think for the different cell type, you have a threshold of how maximum of the mechanical property they can sense. So increasing from 1 kilopascal to the 5 kilopascal, you can see that they actually increasing from the nascent adhesion to become more defined, the cobblestone structures with really defined stress fibers in here. However, when you increase from the intermediate stiffness to really stiff hydrogels, you sort of seem that they don't seem to have the significant differences in cell morphology as well. So again, now we are looking at the cell morphology. Again, then we're looking at the sort of function, looking at how much of the ECM protein the deposition by the cells here. Again, that we grow the cells into the monolayer, and then we're looking at the ECM deposition. The red color, we're using the antibody staining for collagen 1, and then green indicates of the fibronectin. Again, in the fibroblast cases, you can see the direct correlation of the increased stiffness. You have the increases signal for both fibronectin and collagen, which shows you that increase the stiffness, you actually increase more of the ECM deposition, which is onset of the fibrosis, which is being previously demonstrated as well. Interestingly, in the HUX situations, you see almost no collagen signals, as we know that HUX does not produce any collagen 1. However, for the fibronectin, by themselves, and a lot of the signals, we first of all, we don't see any differences between these cells. Second, and then we also see that these fibronectin signals are mostly in intracellular signals as well. So again, we want to show that, even the concept of the stiffness regulates cell behavior, but also that depends how cells sense different cell types, and then in terms of what the range they can sense in the particular applications. So to further demonstrate how we can actually tune the sort of ECM proteins and by signaling and by physicals, we do these sort of myofibroblast activation studies, which is known that to be mostly depending on the substrate stiffness. So we culture fibroblasts on the variety of different stiffness and a variety of different ECM coated surfaces. As we coated these fibronectin on the surfaces and grow the fibroblasts, as you increase the stiffness, you can show the green signal, which is the smooth muscle actin signals positive fibroblast cells, which indicating the onset of the fibrosis. So you can see the direct correlation as you increase the stiffness, you have more cells that indicating that more percentage of the cells actually being activated with the SMA positive cells, which has been demonstrated in other papers. So now when we do actually laminin-coated surfaces, interestingly that regardless of the sort of a stiffness, we'll only see a small increment in terms of how many cells being activated. So now this shows us that, by changing the substrate, how cells are sensing, you can actually control and then dial down that activation of the fibroblast. Then again, we choose another protein looking at is matrigels. Now this is the other end that by matrigel coating, that regardless of the stiffness, even in the softer substrates, you have consistently a high percentage of smooth muscle actin positive cells that have been activated. So matrigels contain a variety of different ECM proteins as well as the growth factors. So you can see that in this cases, you have that regardless of the stiffness, you have most of them all activated. So again, this just shows us that the cell function and behavior, not only depending on the substrates, but also dependent on the ECM substrates they adhere to. So we sort of are closing the studies using a ex vivo studies. So because that we're trying to generate a substrates that culture cells that have not seen any of the being cultured in the tissue culture plots before. So we did these ex vivo studies in which we isolate the cells from the cell skin, and then we're putting these the tissues that on these substrates we make. So interestingly, that we chose the two different models using different stiffness at different ECM proteins, and again we see in the fibronectin coated surfaces you have a directly stiffness modulated cell migration. You can see how these cells are actually migrating through the matrix, the tissues into the substrates. As you increase the stiffness, you actually get more cells migrate. And then not only the long distance is further, but also the population of the cells as well. Now, if you're changing the substrates of the laminin, you see that you have them actually barely adhere to the surfaces, but also have minimum a significant lower number of the cells that adhere to the surfaces. So again, the concept of how to the cell behaviors in terms the response to the stiffness is also dependent on what type of matrix that they adhere to. So for this, I'll conclude my first part of the talk. And the second part talk, I wanted to continue on sort of using these as a platform and then continuing on using the Dextran matrix, then talk about, currently we are thinking about how to breaking the bulk hydrogel into the sort of microgels. As you can see that, traditionally this community has mostly focusing on the sort of amorphous bulk hydrogels that been significantly that dominated for the past few decades. I think about 5 years ago, the paper came out, Tatiana Sugu's lab and also Jason Burdick's lab, they starting to sort of pioneer into the microhydrogels. So when you actually break these small, big particles into smaller particles, you gain a lot of functions. For example, you have increased injectability, the porosity, the bioactivities. You can really apply these different modules by modify the surface with different ligands that can increase the sort of interactions between the concentration, localize the concentration of your bioactive ligands that engage with the cells. You can also engineer in the sort of interparticle interactions. You can also change the different, the density, the shape, the size. You can also, mix and match to do these modularities. So essentially, you not only increase the surface area, which give you a lot of, opportunity to play around with a lot of parameters, but also you can through that you can achieve the modularity and multifunctionalities. So the way that currently literatures are generating these microsphere hydrogels are dominated by the microfluidic systems. So both the microfluidic systems and then the batch emulsion using the same concept, which, you have the aqueous hydrogel precursor solutions that, mix with the oil phases and you vortex them and generating these different particles. You isolate them, you purify them, and you anneal them, form these particles, right? So the batch emulsion is really easy. It's a dirty, quick experiment. You can actually done that. And you do suffer from the heterogeneities in that particle sizes. And then however, when you're using the microfluidic systems, really the beauty is that you can actually control by the speed that you can control these sort of the particle size. But again, you have to go through that purification steps and also the annealing, which can be challenging and compromise the mechanical properties. And then the other, hydrodynamic spraying or the lithography and patterning, you still require the setup, the equipments, and also using, expertise and for that. So our lab is sort of innovating in that kind of a avoid using all the equipments and avoid using organic solvent, and we want to do everything in aqueous conditions. So which we sort of utilize the liquid phase separation as a way to create these phase separation and the particles in our case. So the liquid phase separation is a thermodynamic and kinetic controlled process. So which is you dependent on the concentration, you can actually generate the nucleation or in the spinodal decomposition form these sort of continuous phases, or continue to increase the concentration, you can become the reverse phases, which controlled by the temperature, the soil pH, and the concentration. Of course, you can control either one phase and two phases. So a lot of parameter you can play. And the idea is again that we want to generate a solution that can directly applied to the wound area and close the wound, and then by the body temperature, you can literally switch using the temperature to create these particle formations, and then using that as a guidance for the cells to migrate and facilitate that cell migration to close the wound gap. So this is sort of the overarching of the idea that we had for the lab. Again, that we're using the same material for that case. If you remember in the previous slide, we have these two orange boxes which have the phase separation. So in here, again, that we sort of validating use this sort of a basically heating of the solution. So here we do the basically using cuvette with dextrin solution in the cuvette, place in front of UNH logo. And then if you see the UNH logo, that means the solution is clear. And then if you don't see it, that means the solution become cloudy. So as you can see that native dextrin is hydrophilic, so even increase the temperature, it doesn't show any phase separation. Even low modification, the 40%, you don't see any of the phase separation. At the 70%, which you have to increase the temperature to 48, then you can actually see the phase separation. And then you cool down to room temperature, become clear again. So, and this is of course dependent on the on your modification. As 88%, you can actually create a phase separation at room temperature, which allows us to handling cross-linking hydrogels and handle the gelation formation. Of course, when you're cooling down further to 10 degrees, of course, that the solution become clear again. So now we're using a variety of different techniques to quantitatively analyze it. So here are the dynamic light scattering showing you that at the 10 degrees, you only see the broad peak, that the smaller size. When you increase the temperature to 25, you start to see that splitting of the two peaks have the larger particle forming in the solution. And then when you have increased to 37, essentially all the small peaks are disappeared, and then showing the large peak in the large particle area. And then we also use the UV-vis turbidity assays by looking the absorbance at 400 nanometers. And then we also did its concentration range. And then you can see this is really concentration dependent. As you increase the concentration, you lower the transition temperature when we actually using these as onset, indicating that solution become cloudy. And then we sort of overlay the using the UV-vis and DLS to show the transition temperature. We see the nice a little differences between at the high concentration, but we see in general a good trend as you increase temperature, increase concentration, you lower the transition temperature. So further, you can use the molecular weight to modify control. As you increase the molecular weight, you lower the transition temperature. As you increase the modification, you also lower the transition temperature. We also did a heating and cooling cycle to show this is reversible process. It's a truly an LCST phenomenon. So we using the heating as the red and cooling use heating is the black and cooling is the red, and indicating there's negligible hysteresis, indicating this is a rapidly healing and phase separation and dephase separation process. So indicating that this is a truly an LCST behavior in our system that we've been able to induce. And lastly, we're trying to understand what is the mechanism of this sort of a phase separation in our system. So we know that when you increase modification methodically, you become hydrophilic to hydrophobic residues, molecule. And then when we added the surfactant SDS in the system, even to the 0.01 molar concentration, you actually see there's no phase separation induced at any of the temperature we heated. So indicating that the SDS is successfully dissociated hydrophobic interactions. So we hypothesize that during the phase separation at a high temperature, we have the higher possibility of a colliding of the hydrophobic residues by the methacrylates, which is a driving force for the phase separations. So then we're looking at the morphology. We did the SEM morphology show. Then during the phase separation, they form these sort of defined microparticles, and then increase the temperature. You also have the larger particles. We also test out the cytocompatibility of the particle. Essentially, we incorporate the concentration 10 mg per ml of the dextrin solution in the culture media, which can be phase separate at the 37 degrees. You can see that we culture the fibroblasts on the tissue culture plastic. You can see that these actually phase separate in the well-defined droplets in solution, as well as they don't induce any cytotoxicities to the cells adhere to the bottom of the culture plates, shown here by the live dead staining here. So everything I've been talking about so far for the second project is really on solution. So now we went on to generating hydrogels. utilizing these methacrylate group that we modified to the backbone. So the idea is that you can have the same chemical composition. One is no phase separation, the other one is a phase separated. So essentially we have the same chemical composition, but basically triggering the phase separation that you can create two physically completely different structured hydrogels. So shown here on the top is your phase contrast. In the non-phase separated gels, you can see there's no defined structures. In the phase separated gels, you have these nice sort of droplets, domains that form in these materials. And then the SEM of the non-phase separated gels feature these nanoporosities. And then in the second part, which is the non-phase separated gels, you have these defined sort of particles that embedded. So this is something different from the currently what is the sort of a granulate hydrogel, because those granule hydrogels essentially anneal the particles together. So in our cases, we not only have the particles, but we also have a secondary network. So essentially, we made a composite hydrogels that features these phase-separated domains that crossing within these matrix. So there's a slightly different between the two categories of the materials. And then it is interesting for us to know that actually these hydrogels have these autofluorescence properties, which is great for us to actually observe so that we don't need to label the domains. So again, that no, this is the below cross-linking hydrogel below the transition temperatures. You can see there's no defined structures. And then interesting to us that we actually form the hydrogels at the transition temperature, close to the transition temperature. Instead of getting particles, we have these vesicle formation in the systems, which quite unique and then allows us for future drug delivery applications. And if you continue to increase the temperature, the hydrophobic domains in these cases in the vesicle can continue to squeeze the water was inside of the vesicle and condense. Now we see the completely condensed spheres in these, which is smaller size in the high temperature. When you form the hydrogels. So this is temperature independent phase separated microstructures. We can also changing that with your concentrations. At this 10 mg per mil, you have the small particles. 30 mg per mil, again, you have these vesicles, and then 50 mg per mil, which is a high concentration, you have these multi-domains, but mostly condensed sort of microdomains as well. You have the high concentration, you have a continuous phases as well. So now, to further confirm that it is the hydrophobic, when we see the autofluorescence, we added, in this case, we actually add the fixidextran, which is hydrophilic, and then now red and hydrophobic. So once we wanted to identify if the structures we saw in the early, we actually see this the similar thing. So now in this case, you can see that overlapping with the green and red signals shows you have the yellow inside of the vesicle with the red surrounded the area, indicating that we have truly formed these vesicle structures. And in the 50 mg per mil, we have the continuous phases. You can see now you have completely separate the two domains where you have the highly cross-linking density hydrophobic domains that interspersed by the relatively lower cross-linking regions, which is the hydrophilic side, which is now showing you completely two separate sort of area as well in this sort of 50 mg per mil, which these data confirm the observations we saw in our autofluorescent cases. So now we actually wanted to test out the mechanical property of the hydrogel. So we basically generating hydrogel that non-phase separated and phase separated. And then interestingly, the idea having the particles in the gels, we thought we were able to get a higher stiffness. And surprisingly to us that across the concentrations, we always find that the phase separated gels have a lower modulus, which is showing you in the solid domains here. In the solid squares here. So now, which makes us to rethink about the structure of the hydrogels, why in the phase separated gels have a lower modulus. So now if you're thinking about the cross-linking density in the network, so in the non-phase separated gels, you have relatively uniform distribution in the cross-linking density, which give you higher modulus. But in the phase separated gels, now you have phase separated regions, which are highly cross-linked. So now you have is regionally stiffer, but now overall, because the chemical composition stays the same, so overall you have a softer matrix, but locally I believe that you have this sort of a stiffness, a higher stiffness region. So we're now trying to, this is a bulk hydrogel stiffness characterization. We're trying to using add them to really characterize the local regions of the mechanical properties. And then when we do these 2D cell studies, we actually see that the traditional concept of stiffer substrates will actually promoting a better cell adhesion and spreading. And again, our data shows completely the opposite results. So in the non-phase separated gels, which is higher modulus, we see the less cell area and then less defined focal adhesions. So in here, you see the cells are actually having smaller area compared with the phase separated hydrogels. Now, in the phase separated gels, we use this phospholipid paxillin to show the focal adhesion, and then you can see that in the phase separated hydrogels, actually the cells defined really well-defined a pontate focal adhesion in this case. And if you zoom in, you can see these really on the edge and then over the protrusion on the front and the end of the cell adhesion in this case. So now this seems contradictory to the traditionally established community, but if you think again, it's not the bulk hydrogel properties that control the cell behavior. It is actually the local properties that dominating the cell. So the local environment, the local stiffness is more important to cells to sense. So we believe in this cases, we have the locally that stiffer gels, that stiffer regions, which actually promoting cells behave and spreading better, and then also have these defined focal adhesion. So again, this is consistent with what research reported. However, because the hydrogel is unique compared to the traditional hydrogel, that's why we're seeing this unique behavior. We also went on to encapsulate the cells in 3D by changing the different morphology. So this is very early on our studies that's showing you that in the smaller size, the cells barely actually able to spread. They sort of internalize domains, which we don't really fully understand the cases because we see the overlapping with the green signal, which is autofluorescence of microdomains that inside of these cells, which we don't really understand what's going on here. But then you have the larger domains, which is you can see the cells actually using the interface to really migrate through the systems. So again, that we know that the reviewers asking questions why you can control this, and unfortunately, this is a phase separation conditions. We really can't control really well the phase separation. So again, then we sort of are trying to tackle this problem by introducing the second. Right? If you're thinking about why the domains are actually sort of emerging and become these large domains, essentially when the phase separated regions, they actually merge together and colliding, become growing and bigger. So if we have some secondary domains that actually molecules can separate this, then we're able to control a little bit in terms of their size. So what we did here, we add a small fraction, which is only 10% of the dextran, non-modified, which is hydrophilic, right? So what's showing you is the sugar chain here. If you have a large molecule of the non-modified dextran mixed together, you actually can be successful in separating these microdomains that merging together. You have small molecule together, which is smaller a smaller hydrodynamic radius. It doesn't really achieve the goal because now the particles, the micro domains can still sort of colliding and merging with each other. So we sort of tested this hypothesis and did these studies. And then you can see that as you increase the molecular weight, that you actually able to achieve a relatively controlled, better than before in the domains in this case. So now we're able to using this to sort of as argument and using this to practice cell studies. And then here we show that now you increase the cells, so you have more of the interfaces. The cells can actually sense the matrix and using this interface to really grow. And then by day 14, you can essentially become a completely sort of a cellularized components. You can see that so separated different images. You can see the nucleus are surrounding the matrix. You can see these active filament are surrounding the matrix. So we're able to control that in the system that's really non-degradable, right? In a non-degradable systems, that traditionally you want to have a matrix that's degradable. The cells can pave their way to really migrate. But now if you have this heterogeneity in the mechanical properties and then using these domains as a sort of 2.5D, because it's not truly 2D, it's not truly 3D, so you have some sort of interface. So now you can use this interface and really guide the cells through and migrate and take over the entire matrix. So not only we have this observation of single cells, we're able to show that in the aggregates as well. So here, as you can see that in the non-phase separated gels, the aggregates only show some sort of protrusions to sort of try to engage with the matrix. And then if you have this phase separation, you can see how the cells actually dissociate and migrate and then completely take over and the entire cellular migration. So I think this is really unique for, engineering that sort of a, biocomposite was defined, induced the matrix can actually achieve the signals. So moving forward, we're actually trying to expand the scope of systems. So we are interested in making two-component liquid-liquid separations that trying to, instead of using heparin as a second component, non-modified, as a non-functional component, we're trying to use some other component which has the functionality. So we're trying to introducing heparin, which you can modify the same as the heparin methacrylate and then mix them together. And then we can actually pushing forward in even at 70 degrees, we show at the room temperature we have the phase separation in here. So we can control the modification of phase separation and then also as well as the content, the heparin can really change either the aggregates domains or the reverse phases. So depending on the ratio, this is ongoing work. And then finally we want to show is that we're trying to build these sort of wound models and injured. So these courtesy of the one of the collaborator and colleagues that they developed these sort of injury to tissues on the chip, which now you can generate a tissue and then injure it and then inject biomaterials in the model and using these microdomains and facilitate that migration and closure in the wound healing applications as well. So these are the second part. So with that, I like to conclude and then thank you for the students who did all the work. These are the lab members. The people actually pioneered in these works are Sanya and Rabea, and then the collaborators. And I appreciate the acknowledge the fundings for these applications. And then I'm happy to take any questions you might have in the audience.
Thank you very much for this interesting talk. There's no questions in the chat yet, but I have a couple of questions. First one is about the introduction of methacrylate groups in the dextran. We've done quite a few modifications of different water-soluble polymers introducing methacrylate groups. And what we've noticed that when you introduce relatively large quantity, large degree of modification, the materials, they lose solubility because methacrylyl groups, they are relatively hydrophobic. In your results, you have up to 70% of methacrylyl groups. Did you observe any solubility losses?
So in our cases, we do have a lot of challenges in solubility, particularly on the polysaccharides. So we saw it's dependent on the molecular weight. So for example, the 500 kilodalton is really difficult for us to achieve. So we are able to do anything between 250K and then lower. So we mostly using 86K for our cases, which you can do the 88%. We don't have any solubility issues. We're sort of pushing the boundary because we're looking at phase separation, which is the borderline of solubility issues. But 500 and beyond, we barely can do the solubility issues unless we can really lower the modification in the case. The more challenging we had is we're actually trying to induce other functionality. So because dextran doesn't have any chargers, so we're now trying to balance charge, which is introduced that hydrophility, but also methacrylate or any other, hydrophobicity. But when we actually having secondary modification, that is where we have a lot of issues. So methacrylate is okay, but now we're using methacrylate molecule to another a different functional group that we're having a lot of issues. So that's why I'm here in Virginia Tech to attend some of the glycopolymer conferences to get some ideas how the other groups modify their sugars. So this conference is mostly about sugars. So I'm trying to learn and ask some of the expertise on that part.
Okay, that's great. Thank you. And there's a question in the chat from Mariam Bambakaki. Thank you for the interesting talk. Did you observe any effect of the cell growth on the shape deformation of the microgels?
That's an interesting question. So we haven't truly really looked into that yet. we sort of know the cells like the system really, a lot, which is surprising to us because considering typically any of the polymer that's been either acrylates or methacrylate based polymers, you photo cross-linking it, the network is really tight. So the cells really didn't really migrate anywhere. So we found that even in the interesting in changing that by introducing that domains, the cells seems like the matrix a lot. I think that because in traditional granulated hydrogel, they have the open pores because you have two domains that merge together and anneal together. You have regions in which empty. The cells can actually, migrate through. But in our case, we have two-dimension sort of a network, right? We have biocomposites. So I think the critical part is that we have these two different cross-linking network with the two different mechanical properties. I think the mechanical heterogeneity is a driving force for the cells as they migrate. And then whatever the shape we develop, even in these sort of a weird shape that, globular shape, the cells just engulf and they use this as the surfaces to intentionally are completely sort of a cellularize it and then migrate through. So I hope that answers your question.
Thank you. And next question is from Gokul Kamaraju. And is the concentration of dextran methacrylate solution has effect on the phase separation?
Yes. So we've done a lot of concentration range. So for the most ones, the lowest concentration to have an effect that we need to be 10 mg per ml, which is lower. So in that you can trigger a phase separation, but not fully form gels. So right now, the lowest concentration we're able to form hydrogels are 20 mg per ml. That's also depending on the modification molecular weight. But 30 mg per ml, I would say, would be um, a good way for that. Yeah.
Okay, thank you. Next question is from Ahmed Moran. Excellent work with an enthusiastic speaker. Thank you. Liquid-liquid phase separation also produces two phases with different polymer compositions. How does this relate to mechanics?
That's a great question. So my second part is we're only looking at a one component. My second part of the work, we're introducing the two components, right? So we have the dextromethacrylate, we have the heparin methacrylate. So these two polymers, we can label the different polymers by different fluorophores. We can observe the different their distribution in the network, and then we can tune the different portion. As a heparin, we know the heparin would be difficult to achieve the high modification. Right now, the highest modification for the heparin is 20%. So that's a lower components. So the dextran, we have a variety range we can do. The heparin, we won't have that much opportunity. But the heparin, the nice part about that is that they're highly negatively charged. So we can further drive that phase separation. So not only having the second component that can contribute to the network, but also they can further drive and then easily for us to form the unique domains. And with a higher density of the region, I believe we should be able to achieve that spatial differences in the mechanical properties. Not only we have two components, but we also have very unique molecular properties of the two different components.
Okay, thank you. And another quick question from Sergey Filipov. Urea disrupts hydrophobic interactions. Did you consider using it instead of SDS to figure out the nature of interactions?
Yes, that's a great question. We actually also use urea that in our the paper that we published. So urea, we haven't seen that's effective. Urea, we have to increase to at least a 4 millimolar concentration to able to show the increase of transition temperature. It does to dissociate the hydrogen bonding and then also the hydrophobic domains in actions. So it's not as effective as SDS. As a matter of fact, we actually just one paper that we recently submitted that on the review right now. We're looking at the entire categories of different surfactants. We're looking at about 6 different surfactants, which are different micelle concentration, also the HLB values, as well as the charge properties. And we see that not only different value with HLBs and different charge densities also have a different abilities to make the different unique structures of these hydrogels as well. So